Good morning. You are in tune with Murmu Biology, and we were looking into lecture series of Jharkhand Eligibility Test Life Science Detailed Solution. We were seeing question number 65, and question number 65 says in eukaryotic expression cloning, inclusion of internal ribosome entry site IRES allows. Options are DNA replication in mammalian cell, translation initiation dependent of 5' cap, efficient intron removal, and nuclear export of mRNA. Okay, let's begin. So this is the question. So in this question, there are different terms that I must be explaining uh, in brief so that the question becomes crystal clear to you. So the terms are eukaryotic expression cloning, inclusion of an internal ribosome entry site IRES, DNA replication, translation, efficient intron removal, and nuclear export of mRNA. So let's see what these are. So if you have seen question number 63, the term cloning was there, isn't it? The cloning experiment was uh, revealed in that, and it has been discussed, but let me explain once again. So in molecular cloning, what is a molecular clone and what the vector or a plasmid vector is, isn't it? So anything that is a copy of DNA, isn't it? A DNA copy or genetic material copy that has been transferred to the cloning vector or the transport department type of thing, that is known as molecular clone, isn't it? Any two cells which are similar in genetic information or DNA are known as molecular clone, and how this is done is what we will be seeing. So cloning vector, we take a cloning vector. uh, Cloning vector are naturally found in bacterias and prokaryotic organisms. Okay, and we take gene of interest. What like say let's say insulin, we want to produce insulin gene or insulin protein in the bacteria, so that we can uh, get benefited from it. So we cut the insulin gene. Here the red dot represents the insulin gene and the vector is also cut, then both are ligated together, isn't it? And this vector is then reinserted into a bacteria so that it replicates itself and it produces insulin, isn't it? So this is the molecular overflow. This is important that you understand this. So the in step number 1, the isolation of insert DNA, that is the, let's say here we have taken as an example insulin gene, and it has been amplified. That is, it has made multiple copies of the insulin gene, and it has been inserted into the plasmid vector, the vector which we have obtained from the bacteria, and both are joined together and inserted into the bacteria with the help of many uh, processes. It may be heat shock, it may be electroporation, it may be gene gun, anything. And then it is cultured, and it is tested for the successful of the experiment, and then the bacteria is further downstream processed to produce insulin, isn't it? So this is a molecular cloning workflow. You must understand this. Okay, now that you have understood what molecular expression cloning is, let's proceed to another. For a time being, I am omitting option number 2, that is IRES. You will get a clear picture once you understand the molecular processes. So these processes are discussed now in every cell, isn't it? The central dogma, the central dogma is the replication, and replication again, from replication the uh, transcription takes place. After transcription, the mature RNA is brought inside in the cytoplasm, that is brought about in the cytoplasm with the help of certain export protein. from the nucleus, isn't it? It is brought in the cytoplasm so that it undergoes translation and ultimately the protein is being made. So this is the central dogma of life. If simplified, the DNA, from DNA the RNA is made, that is known as transcription, and from RNA the translation is takes place. And when DNA itself is replicated, DNA itself is made, itself copying, then it is known as replication. So these are the overview, isn't it? DNA replication takes place, and it's in replication the number of DNA is doubled, isn't it? DNA replication generally takes place before, when the cell is about to divide to distribute the DNA. The DNA replication takes place, isn't it? Transcription takes place. So in transcription the mRNA is made, isn't it? mRNA means the code through which the protein will be translated. Protein, what is protein? Every structural form in the cell is an example of protein, isn't it? So these are the basic, you already know that. So during transcription and translation there is many events, isn't it? So one of the event is intron removal, isn't it? You know that non-coding sequences are known as non-coding sequences has to be removed from the premature mRNA, isn't it? So all introns are being removed and the exon only comprises of the mRNA, isn't it? This is known as splicing. So this is a simplified version. Transcription is there and mRNA comes out of the nucleus, and the translation takes place in the cytoplasm. Okay, so these are normal processes, isn't it? So we will dive more into translation. So here you can see in translation there are three steps. There is initiation, there is elongation, and there is finally termination. You can see in initiation the initiation factor comes along the mRNA, and it scans throughout the mRNA to find the initiation codon AUG, isn't it? You just have to, don't have to go, we will go in detail in another times, but you just know the concept that when ribosomes come before translation, when a small unit of ribosomes come, which is brought from with the help of initiation factor 3 and 4 and 1, they bring the small subunit of ribosome to scan the mRNA, isn't it? They scan the mRNA and search for AUG codon, isn't it? When they find the AUG codon, then the large subunit of the ribosome comes and attaches, and the elongation process continues. In elongation process the protein is being made continuously, and finally the termination takes place, in which the final protein is being produced, and then the protein does its own function, isn't it? So in eukaryote this is the step we are searching for, isn't it? The small subunit is being brought by initiation factor 4. It scans the mRNA and searches for AUG codon. Here you can see that in step number 3 it has found the start codon, isn't it? And then the translation begins. So this is the general process. Now when you just remember that you have just seen here that in molecular cloning, isn't it? In molecular cloning the vector brings about the gene of interest to the bacteria, isn't it? So in this case there was insulin gene, and in this case the IRES is present, isn't it? In case of insulin gene, this IRES DNA sequence, RNA sequence rather, is present in the vector, and that RNA sequence is added to the normal gene. So IRES is nothing but a piece of RNA, isn't it? That has been brought about with the help of vector to get attached to the genome. And where this RNA IRES, that is RNA element, joins, then this automatically brings the ribosome, small subunit ribosome and large subunit ribosome, and it starts the translation. Okay, so what this RNA, what this in case of uh, IRS independent, that is in natural translation, we have seen that the small subunit is brought about by the initiation factor, and the initiation factor scans for the EUG start codon, and then also in between the large subunit gets attached to it and the translation starts. Okay, so if the ribosome entry, IRES entry, is there with the help of vector, then what this IRES does, this RNA automatically recruits small subunit and large subunit, and it automatically starts the translation. So this is the whole concept of this question, isn't it? So in natural translation, this you can see that a small subunit is scanning like an aeroplane in a large runway, isn't it? And in IRES, it is like a helicopter, or it just lands on the uh, on a specific place, isn't it? So here you can see the comparison of natural translation and viral mechanism for translation. This IRES is naturally present in the viral translation, isn't it? So natural translation, the small subunit scans the mRNA, isn't it? Somewhat like the aeroplane running on a runway, isn't it? And the in IRES mechanism, the IRES is just attaching to the specific place, and it starts the translation. So this is the concept. So now you have learned about eukaryotic expression cloning. You have learned about what IRES is. You have seen what DNA replication is. You have just seen what translation is, and efficient intron removal splicing, you have seen what splicing is, and nuclear export of mRNA, you know what that is. So this is the reason. An internal ribosome entry is a specialized RNA sequence that allows for translation initiation in an end-independent manner. That is, it is not dependent on anything, isn't it? So when a small RNA is brought about, it finds for the M7 cap in the mRNA and then scans, isn't it? The 40S ribosome, which I am talking about. Option number A is DNA replication. Replication is not related to this, because IRES is related to translation. So this option is somewhat wrong, isn't it? Intron removal is also another process, RNA splicing, and is mediated by spliceosome. So this is also non-relevant. And nuclear export, mRNA export, is directed to the export protein, isn't it? So these are the literature of why option A and option B is correct. Okay, so you can read this. And option C is also incorrect. Why efficient intron removal is RNA splicing, as we have just discussed. And nuclear export, you know what the export protein are responsible for. This is also irrelevant. So you now know the correct answer. In eukaryotic expression cloning, inclusion of an internal ribosome entry site, that is IRES, what it is, it allows translation initiation independent of 5' cap. 5' cap is related to the mRNA that is found by the small subunit of the ribosome in case of natural translation. And in IRES, the IRES automatically gets attached to the mRNA, and it starts the translation. Okay, so you can support us also, and if you cannot support us, you can help us root and grow with us with liking the video, subscribe and share, and download for offline viewing. So thank you for your time and stay tuned to Mormo Biology.